Hello students, today we will start with another type of cross, that is trihybrid cross. In trihybrid cross, we read about the inheritance of three characters combinedly. So, it is the inheritance of three characters. Inheritance of three characters. Now here, total number of offsprings. Total number of offsprings are 60. We have seen that in monohybrid cross, the total number of offsprings according to the parents that we have taken is 4. In dihybrid cross, the total number of offsprings that we have seen are 60. And here we have taken consideration of one character. In dihybrid ratio, we have taken consideration of two characters. In the same way, in trihybrid cross, the total number of characters that are taken are 3 and total number of offsprings obtained are 64. Now, in monohybrid cross, we have seen types of gametes are 2 and total number of gametes are 4. Here we have seen types of gametes are 4 and total number of gametes are 8. In the same way, here types of gametes involved in F2 cross, F2 generation. Types I have written here. Different types of gametes are 8 in number and total number of gametes for F2 generation. When we are framing out the Punnett square R16, when we write a Punnett square like this, we used to represent first 8 number on the top area and on the left side we used to represent another set of 8 number. So total number are 16. Now here, what are the 8 types of gametes that are involved for the F2 generation? If we come for this one, first 3 types of characters. Let's take 3 different types of characters as capital AA, BB and CC. We should consider that each pair of allele is responsible for one character, but all the 3 are combinedly getting inherited to the offsprings. Now, if this is crossed with the recessive one, like this, then 8 types of gametes from this one obtained will be, from here the 8 types. From here we will get ABC one type, small ABC one type. This is from the parents. Now, in F1 generation we will get the type of genetic combination like capital A small a, capital B small b, capital C small c. Here we have written for F2. For F2 from F1, the 8 types of gametes are, from this I am going to represent capital A, capital B, capital C. Now, capital A, small b, capital C. Another, capital A, capital B, small c. Now, capital A, small b, small c. 4 types. Another one, from small recessive allele. Small a, capital B, capital C. Small a, small b, capital C. Small a, small b, small c. Now, small a, capital B, small c. So, 8 different types of gametes that we have seen after F1 to obtain the F2 generation. Now, when these are represented on the topmost area of the Punnett square and on the left side of the Punnett square, obviously the total number 8 plus 8 will be 16 number of gametes. Now, if we want to calculate the phenotypic ratio, if we want to calculate the phenotype ratio of trihybrid cross, already we have seen in dihybrid as 9:3:3:3:1. We can just multiply with one more cross, that is monohybrid phenotypic F2 generation ratio. So we will get a ratio like 27:9:3:3:3:1. You will get the ratio like this phenotypically. In the same way, if we multiply the dihybrid genotypic ratio with again one more extra ratio, that is 1:2:1, which we have seen in monohybrid cross, then we will get some different types of ratios like this regarding the genotype. Test cross is done. This cross is done to know the unknown genotype. Of a parent. To obtain or to know the knowledge of the unknown genotypic of the unknown genotype of the plant or parent, the unknown is crossed with homozygous recessive genotype alleles. Now, we know that homozygous recessive, in the sense, it should be represented with small letter alphabets. If we write with capital letters anything, it indicates that we are talking about dominant alleles, homozygous dominant alleles. So, first possibility, there are three possibilities to explain. So, first possibility, suppose take an unknown. It is crossed with homozygous recessive one. Suppose this unknown is having the genotypic allele like this homozygous dominant. Then in F1 generation, we get all are phenotypically expressing the dominant character, that is top. So we get the allele forms like this. It indicates we get all heterozygous alleles in offsprings. So here the ratio will be phenotypic ratio. If we draw the same thing in form of Punnett square, dominant is to recessive, that dominant character is tall, is to recessive character dwarf. Then the ratio is all are tall and zero number of dwarf plants in the F1 generation. So this is one possibility. Then when in the F1 generation all are expressing the dominant phenotypic character, then we should understand that the parent or the unknown that we have taken is homozygous dominant. Still there are some exceptions like incomplete dominance. We are not talking about that. We are just talking about the type of alleles and the genes that are purely expressing the dominant character. This is second type of cross. And the unknown is having internally heterozygous type of alleles, but think that we don't know about those alleles. Now we have crossed with homozygous recessive parent. Then what happened in the F1 generation? In F1 generation, when we cross this one, capital T is crossed with this one, but still think that this is unknown. But according to our just as imagination or estimation, draw a Punnett square and check. Suppose if the unknown genotype is having alleles like this, and we have crossed with homozygous recessive one, then we have got one number of this homozygous recessive we got heterozygous dominant, again homozygous this one. Now what is the number? Heterozygous dominant one are 2 in number that are expressing the dominant character. Homozygous recessive are 2 in number. Now if we write, we can say we have seen 50% of dominant phenotypic character, dominant phenotype, the dominant phenotype, that is we can call tall plants. Now we have seen, we should write like this after so that as a student we will understand this type of answers also. 50% of recessive phenotype. Recessive phenotype. What are recessive? Homozygous, this one, dwarf or a homozygous recessive type of expression in the offsprings. Then it will be dwarf plants. Dwarf plants. If we write a phenotypic ratio of F1 generation, phenotypic ratio of F1 generation, we can write like tall is to dwarf. We can write 2 is to 2 because the number of obtained offsprings with the dominant allele are 2 in number. That only we are writing here and expressing in the phenotypic ratio also. The third type of cross is again unknown. Suppose, think that this unknown is having the alleles like this, but we don't know exactly. After crossing and after seeing the F1 generation, based on the phenotypic characters of F1 generation, then we will understand or we can analyze what type of unknown genotypes might be. So here, again draw the Punnett square and let's check. Suppose think unknown is this one and we don't know exactly initially, and the homozygous recessive one, that we know that we are taking that type of plants only. If we cross, then in F1 generation we will see all are of a dwarf. Then we will understand that the unknown taken parent is having completely homozygous recessive type of alleles, when all the offsprings in the F1 generation are of recessive phenotypic character. Now, as all the offsprings in F1 generation represented or appeared as complete dwarf plants, we can conclude that the unknown might be of homozygous recessive, that is recessive genotypes. Recessive genotypes. Now, let's see some more points regarding this test cross. When we have concluded that the first possibility has given all tall plants, when we have crossed this unknown with dwarf one, the ratio is 4:0. So here ratio is 4:0 in F1. Second possibility, when we have seen the unknown crossed with, we have to consider this as unknown, which I circled. the different types of genotypes or alleles. Here we have got 2:2 phenotypic ratio in F1 ratio. By this heterozygous type of alleles crossing with homozygous recessive, we should give one conclusion that ratio of both genotypes and phenotypes are equal, that is 2:2. And also one more cross. Here we have got all F1 offsprings as for this one heterozygous tall with heterozygous different types of genotypes. Now here also we can tell that both the phenotypes are tall and all genotypes are of heterozygous. So genotype and phenotype are equal here. Here also the phenotypic expression is 2 tall and 2 dwarf, and the genotypic expression also is the same. 2 different types of alleles. Here also 2 same type of alleles. Even in the third possibility, when we have taken this cross and crossed with the known, already we know the genotype of the plant that we are going to cross with. Here also we got the phenotypic ratio at 0 tall and 4 are dwarf. So here also we can conclude that the phenotypes which are responsible for tall are not obtained, but all the offsprings are having completely homozygous recessive genotypes, since all showed phenotypically dwarf nature or recessive nature. That one I will conclude finally. In all types of crosses, the phenotype number and genotypes number we can tell or a ratios. Simply the phenotypes number and genotypes are same. Here 4 are tall, 4 are having heterozygous. 2 are tall, 2 are having heterozygous. 2 are dwarf, 2 are having this one. Okay, all the genotypes and phenotypes are same in all the 3 different types of crosses. Here instead of this one, sometimes they use 50%. Then also we should be able to identify what type of cross the examiner is going to ask. Next, back cross is done to increase the superiority of the offsprings by crossing with homozygous dominant genotype. Here also there is no point of taking the unknown. We can take any of the known genotype or unknown genotype, whatever it may be. So here we can cross any type of genotype. Here any type of genotype from a parent or from F1 generation or from F2 generation, and we can keep on taking whatever the genotypic may be. Generation. We can take any of the genotypic alleles and we can cross with the pure dominant one. Is crossed with pure homozygous dominant alleles. Example, it can be capital T, that is known, again crossed with this one to obtain the pure lines or true breeding. Another one, we can take heterozygous one and we can cross with pure homozygous dominant alleles. We can take the recessive one, again we can cross with pure homozygous dominant alleles. Now we will see the crosses, different types of crosses, and the ratios what we are getting here. All F1 offsprings are of tall phenotype or tall character or tall trait. Now, what are those tall? With genotypically expressing this type of character. Here, the ratio. What ratio? Phenotypic ratio. Tall is to dwarf. Here, ratio is 4:0. Second possibility, heterozygous known. Again, cross with pure this one. Again, draw a Punnett square and check what are the types of alleles and what is the phenotypic character. In F1 generation, the phenotype, or since we have used in the definition, we can take any parent or F1 or F2, we can write in the next generation. Ratio will be 2 tall is to 2 dwarf. 2:2 ratio, because 2 are of purely tall. These are also tall, but phenotypic ratio we can take, tall is to dwarf, all are tall. Again here it is 0. Next. Now, what is the genotype here? Genotypic ratio. 2 are of homozygous dominant, 2 are of heterozygous dominant, then answer is 2:2. Genotypic ratio is 2:2 here. Phenotypic ratio is all are expressing tall, because though the recessive allele is there, it will not get expressed, it is suppressed in presence of the another type of allele, so we are calling it as dominant one. Here also, if you see, all are tall plants here. Next, third possibility, if you take like this, then if you draw the Punnett square, small t, capital T, capital T. So the ratio, if you take, 2 are tall, yes, there is no, all are showing 4 are tall. 4 are tall. Here also, if you write the genotypic ratio here, then it will be good, I think. That is here we can write this is of tall, this is of dwarf. Okay, so now we can write some ratios here under this. Here 4 are tall in the sense all are heterozygous dominant alleles genotypically. Phenotypic ratio is all are tall, no? So tall is to dwarf. So it will be 4:0. Coming to genotypic ratio, that is capital T, there is no point. Capital T, small t is there. Capital T is not there. Small t is not there. So these are 4:0. In any of the cross, there is no point of existence of a dwarf phenotypic character or the alleles that are responsible genotypically for the dwarf character or dwarf trait or dwarf phenotypic expression. Here it is 0, here it is 0, and here also it is 0 regarding the homozygous recessive alleles or phenotypically dwarf type of character.